Hi, everybody. It's Merm. It's hard to believe. Oh my gosh, it is week 4 of this, which means we are on to unit 4, which is really about how cells communicate and reproduce. So, a lot of cell signaling, signal transduction pathways. I want you to think back to when we acted it all out and did like the role play of how does your body get signaled by epinephrine to change glycogen into glucose. That's a great example of a signal transduction pathway. So we start off here with cell signaling in order for an organism to produce meaningful responses to stimuli. Basically, if you can't respond to stimuli, you're not going to survive. You're not going to be a good cell. So cells and organisms as a whole have to be able to coordinate their efforts and respond to changes, whether it's internal or external. Cell communication is necessary and must be regulated and specific. That means you have to have a controlled response to something to react. And the signals, we call those ligands. And ligands come in several different forms. Ligands could be proteins, they could be steroids, they are usually like hormones, neurotransmitters, things like that in your body. They can be within a cell or in the external environment. And within an organism, only target cells. So this doesn't have an impact on every cell in your body, but instead it's very specific so that a target cell will respond to a stimuli. They have the right receptors. So it's all about the shape, the ligand. Let's say it's a protein and it has this totally unique shape. Well, there has to be a receptor on the cell that can actually bind to it and be perfect for it. So here's my cell, this is my receptor, and this is my ligand. All right, direct contact versus signaling. So signaling is when you have a ligand that is released in one area that targets another area. All right, so this is a great example. This molecule could have been traveling through your blood and then it's picked up by a receptor and it works. So a great example, like in reproduction, your eggs are only going to be stimulated to go through ovulation when a follicle stimulating hormone from your brain targets the ovary. And that follicle stimulating hormone is going to travel throughout your entire body, but it only has receptors in the ovary, so it only works on follicles. Direct contact is cell-to-cell interaction. And so I think about cells having direct contact with each other. And this happens a lot in your immune system. Your immune cells will actually have direct interaction with another cell in order to tell it like, hey, this is the invader. So we think of like you have B cells, and B cells are able to make antibodies, and you have macrophages. And macrophages are able to destroy something and then show the markers of what they destroyed. So these are like specific little chains on their cell that are antigen markers, and they're able to show what was the antigen that they digested. All right. There are many examples of cells communicating with each other. So I went through several. I really like showing you diagrams like this because the AP loves these diagrams. So take the time to really look at the diagram and digest it. So first of all, this is a specific example. The AP is not looking for you to like know what a helper T cell. That in particular isn't a part of our curriculum, but to be able to apply biology that you understand to how our body works, and using an example like helper T cells, they would describe the example and then have you explain how that works. So a helper T cell is part of your immune system. And so we look down here and we find where it all begins. So it all begins here with this arrow. So a bacterial infection. So bacteria have gotten into your body, and the bacterial infection caused this inactive caspase-1 to become an active. So they activate it here. And this activation causes two more things. So it's very often a chain reaction. So this chain reaction is causing this terminal trichodermis or gastroderm, whatever this is called. It causes it to open up these pores. So it's causing something to happen that is going to increase pores in the cell membrane. And the actual terrible thing about that, if you think about it, a bacterial infection causes your cells to open up and have holes in them. And the holes might let out things like pus and stuff like that from a cell, something that it is destroying. And over here, it shows that it's making these active interleukins. Well, interleukins are chemical messengers that are sent out to other cells. So basically, your immune system responds by making these chemicals to go signal other cells and say, look, we are going to have to increase B cells. We're going to have to increase T cells. We're going to have to launch an immune response because we are being attacked by a bacteria right now, and we've got to take care of this. All right. Protein hormones. So protein hormones are those ones like that I showed, that they are the ligands, and then they are received by a receptor. And so a great example, follicle stimulating hormone is made in your pituitary gland up in your brain, but then it targets your ovaries. And your ovaries will go through ovulation and will release an egg. And so the releasing of the egg from the ovary is only stimulated by this hormone, and the hormone is made somewhere else. So protein hormones go through the blood and target very specific cells. They have a specific shape because they're a protein. A steroid hormone, as soon as I see steroid, I'm writing nonpolar. These are able to cross the cell membrane without any trouble. The polar tail or polar head and the nonpolar tails let it right through. So they're able to cross the membranes. So these have intracellular receptors. So let's think about an example of that. Testosterone. Testosterone is a steroid. It is a sex hormone. So it's able to go right into a cell, and then the receptor is in here, and it usually causes a change in transcription factors, binds to the DNA inside the nucleus, and causes specific proteins to be made, like facial hair. All right, moving on to the next one. All right, another example, neurotransmitters. So I think you will remember from baby bio, here's one nerve, and then here's the next nerve. And the space in between it is called a synapse. And since the cells aren't in direct contact, you have to have a chemical messenger. So inside of here are all my little neurotransmitters. And when an impulse passes down through it, it will release them into here. And then this next neuron has to have receptors on its dendrites to pick up the messages. So these pass from one neuron to the next to send an impulse. And let's think about pain medication. You take pain medication and it gets right in here and it blocks it. And so it can actually go in these receptors and block the receptors so that the neurotransmitters aren't picked up. And that helps you because if you're not picking up the impulse, this pain message that your body is trying to send saying it hurts, it hurts, it hurts, something's wrong, and you're like, yeah, I just had surgery, I know something's wrong. It will stop the pain message because it blocks the neurotransmitters from being received. All right, signal transduction pathways, 3 steps: reception, transduction, response. Always in this same exact order, always in order: reception, transduction, response. So reception is all about ligand binding to a receptor. And so it is very, very shape specific. And it will only bind to target cells because target cells have the right receptor. They cause a change in the receptor. So a great example to talk about is a G protein coupled receptor. So if this is my cell membrane and this is my receptor. A G protein one has a little subunit coming off the bottom of it. And as soon as a ligand bonds, this actually moves. And so when it moves, it starts transduction. It starts this pathway in order to have a response in your cell. So transduction is the intracellular, so inside the cell, intracellular pathway. We usually think of it as a cascade, and the main goal is to turn on kinases. Kinases are regulatory molecules that are normally inactive, but they get activated by adding a phosphate group. So phosphorylation cascade is a really common term that you hear, and it's part of the transduction step. And that usually involves secondary messengers. And secondary messengers, the most common one in our body is cyclic AMP. So if I look back at this reaction, I could have activated an enzyme like adenylyl cyclase, which will make cyclic AMP, and cyclic AMP will turn a kinase on by phosphorylating it. And then that will turn on another one, and another one, and another one, and another one. And this is all still within my cell, but that is my phosphorylation cascade. The part of it having multiple steps give you multiple chances to turn it off. Multiple steps equal regulation. So if you realize like, hey, you know what, I really don't want to do this, you can actually abort mission, like stop doing it at some point along the way. All right, the response is your result. What happens because of your signal transduction pathway? And so your cell might release a chemical. Like maybe it's going to release glucose. Maybe you got scared and now you have to increase your blood sugar level, and that's how you're going to change glycogen, your stored energy, into glucose, your monosaccharide. Or maybe you are going to turn on a gene. And if you turn on a gene, you're going to do transcription, which means you're going to make messenger RNA. And that messenger RNA is going to go out to a ribosome, and it is going to make a protein. So that response is to make the protein that your body needs. Basically, you've got all this DNA with like a million directions to make so many different proteins, but you only make proteins when you're signaled. So the result of the signal transduction pathway is to turn on that gene and make that specific protein that your cell needs. All right, so on the next page, I have a bunch of examples, diagrams. AP Bio loves these diagrams. So here is my cell. This really shouldn't be called a wall. This is really a cell membrane. And here's my cytoplasm. So step 1 is reception. Step 2 is transduction. And step 3 is my response. So here is my ligand, and here's my receptor. This is my phosphorylation cascade, the turning on of kinases. And then over here is my response. So maybe I am going to make a protein, or I am going to release a sugar. There's like literally endless things that my cell can do based on what cell it is and where it's located and what its job is. All right, and here's another picture. And so step 1 is my reception, and step 2 is my transduction, and step 3 is my response. So always in the same order, always recognizing those pathways. So here we've got 2 different ligands that are happening. The first one is right here as light, and so light is causing this phytochrome to be activated. Again with the wall, this should totally be membrane, unless it's like some bacteria or something, but I think they're showing human cells here. But anyways, here's my light, here is my receptor inside of the cell, this is my kinase, and so it is being activated. And when it's activated, it forms a transcription factor, and transcription factors go, and here they are phosphorylated. That means that they're active, and that means they are turning on my DNA. When they turn on my DNA, they are going to transcribe it, and they are going to do, so we're going to do transcription here, and we're going to make messenger RNA that is going to go out to a ribosome and make a protein. So this is definitely going to be my response. And what happens is, see how there's multiple pathways? Very common in cells. It's not so simple. We're turning on multiple pathways here. The light activating this receptor also opens this channel. So channels allow ions to enter or exit a cell. They're always moving high to low concentration if they're going through a channel, because that's facilitated diffusion. And so here, my calcium, now the gate is open, calcium comes rushing in, and calcium helps to bind this transcription factor and allow transcription to occur. So this pathway had multiple steps. All right, terms. There is definitely a lot of terms here that I want to make sure that you recognize and are able to use. So ligands are our signal molecules, and they definitely have a specific shape, and they bind to receptors. They very often, unless they are a steroid or some other nonpolar molecule, they very often are proteins, which means they do not enter the cell. They cause a change in the cell because the receptor changes shape when it binds to it. All right, a kinase is a regulatory molecule, and it is activated by a phosphate group. And so when you add a phosphate group, now it can go turn on enzymes or turn on transcription factors to allow a response to occur. And a phosphorylation cascade is multiple kinases, one by one, activated. All right, and again, the thing about a cascade that's pretty cool is that it has a level of regulation. It is not going to just give you one change, one response. It'll have multiple steps so that you can turn off if you need to. Amplification means that you amped up the signal. So one signal molecule might cause the release of many glucose. So all you needed was that one little bit of epinephrine, and your body released so much glucose it increased your blood sugar level like crazy. So you can amplify a signal, meaning you can make it multiply. Types of responses. We have done this already, but really common types of responses are to release a certain chemical like glucose or something that it made, or to turn on a gene and make a specific protein. There are many others though too. You might cause the cell to die. The cell could be infected with something, and the signal transduction pathway tells it, like, let's go through apoptosis. Like, let's go ahead and kill ourselves because we are not helping this body any longer. We are a factory for this virus or bacteria, and all we're doing is growing it. So the response could be causing the cell to die. Or it could be causing the cell to increase its permeability and allow certain chemicals to enter or exit the cell. All right, example of epinephrine. So this was the one that we actually did in class. This was the one that we acted out. So we started off with our receptor, and our ligand was epinephrine. So epinephrine is a protein ligand, binds to the receptor. The receptor has a G protein here, and when it is activated, this moves away. And when it moves away, it goes to an enzyme that is in the membrane of my cell called adenylyl cyclase. Then adenylyl cyclase makes cyclic AMP. That is my secondary messenger. My secondary messenger turns on kinases. So here's my kinase. It gives it a phosphate group, which means that now it is active. And that usually activates another kinase, and it does this whole phosphorylation cascade. Turn on, turn on, turn on. Now, eventually, this activates an enzyme, and the enzyme is called glycogen, because that's what it works on, phosphorylase. And it's activated, so it has this little phosphate group on it, and this enzyme takes glycogen and when the enzyme works on it, the enzyme does cleavage, it does hydrolysis, which means it's going to break all of these. And so then I have all of these glucose molecules, which then move out of the cell, and that's going to increase my blood sugar level. So that is my example of a signal transduction pathway. And let me just go back up here, and I want to show you this is step 1, that's reception. All of this here is step 2, that's my transduction. And then down here, when I release all this glucose, that is my response, which made my blood sugar level go up. All right, really important that I always have all of those 3 steps. All right, and I have some more diagrams. Oh, what is apoptosis? So apoptosis is programmed cell death. I'm going to give you 2 examples of where this happens. So in utero, which means before you're born, between your fingers. So look at your hand for a minute. If you saw pictures of an embryo, your hand would look much more like this. And what happens is the cells start to die here, 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 here. Oh, I only gave you, oh no, I gave you enough. All right, and so when they die there, then you're able to have distinct fingers. And if they did not die, if the cells were not signaled and they were not programmed to die, then you would have webbed fingers. So apoptosis is really important there, but it is also important when a cell is infected. So if you have a cell that is infected, it often becomes a factory for making whatever the virus or the bacteria that have infected it. And so these will often cause your immune cells to send a signal and say, you know what, enough. This cell needs to stop being a factory for this invader. And so the cell dies. And so apoptosis is the signaling pathway causing a cell to not live anymore because it is not helping the body that it's in. All right, this diagram is showing, here is my step 1, there is my receptor, there is my ligand, here's my inactive protein and it's becoming active. And you can actually see the phosphate group on all of these kinases. So these protein kinases are being turned on. So this is my secondary messenger. This is probably cyclic AMP here. And so it's phosphorylating. So here's my step 2, my transduction. And then my response is to make an active transcription factor, which is going to make messenger, oh, it's already written right there, messenger RNA, which is going to go out to a ribosome and make a protein. So just another drawing, but really good because AP loves giving you these diagrams and having you analyze them. Oh, all right. These receptors are super cool. They are called tyrosine kinase receptors. It's probably written on here somewhere. Yeah, receptor tyrosine kinase proteins. So when they're inactive, they look like this, 2 separate molecules. And what happens is this little ligand binds, and when 2 of them bind, it forms what's called a dimer. It comes together. And then each of these little tyrosines can get activated by ATP. So you can add a phosphate group. See these little phosphate groups added to each one of them? That is what is going to make this active. And when it is active, each of them might bind a different molecule, and each of those cause a different reaction to happen. So you could have multiple reactions from the binding of this one tyrosine kinase receptor. And I think I told you before that I had a student, Jen Lee, who went to Princeton, and she studied tyrosine kinase receptors, and she studied how the body is affected by light levels and how that is actually activating tyrosine kinase molecules in your body. All right, next one. altering signal transduction pathways. So if you have this pathway, boom, 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 and you get this response, and that's normal in your body, what could make it go wrong? So a mutation can change the shape of the receptor, because the receptor is a protein. So if you alter the DNA, you will alter the protein that you make. So if your receptor used to look like this, and then it is altered and it looks like this, it might not work anymore. This is not going to fit in this space anymore, so it doesn't work. Medications. So a medication would do this. It would have the same shape, and it would come in here and it would block. So usually pharmaceutical companies are always working on figuring out what receptor causes this pathway and how can I make a medication that would block the pathway and not allow the ligand to bind there. And then toxins might also do the same thing. Here we see a picture, and these little antibodies are blocking the way. So here is your muscle, here's all the receptors, and here are the receptor molecules. So we have a nerve coming in here, and the nerve is trying to activate the muscle. And so the neurotransmitter that passes between motor neurons to a muscle is called acetylcholine. So each of these little triangles are my acetylcholine. But right now, my acetylcholine can't fit in my receptor because of these antibodies. So these antibodies are blocking the way. So in this one, you are ending up getting no muscle response. And that's because the antibody is blocking the receptors. So I'm hoping that you are getting really good at analyzing drawings like this and seeing what's going on. All right, feedback mechanisms. So feedback can be positive or negative. If it's positive, you make it go up, up, up. If it's negative feedback, like, oh, this is getting too high, let's bring it back down. Oh, it's too low, let's bring it back up. And so these usually fluctuate around what we call a normal level. So I took this little diagram. Well, actually, let's just write this here. Homeostasis is our internal balance. And so negative feedback is what fluctuates in response, and it brings it higher or lower. So I'm going to put blood sugar level. So you think about like, hey, I just ate something. Your body's like, whoa, way too much sugar. Let's make some insulin. Let's store this sugar. We just have way too much of it. And then you can, if you have not enough of it, you can break glycogen into glucose. All right, here I want to look at this one. This was on an actual AP exam. So your hypothalamus is an organ in your brain, and so it makes TRH, and that affects your anterior pituitary, which is actually right next to it, another organ in your brain, and that makes thyroid stimulating hormone, TSH, which stimulates the thyroid to make thyroxine. Thyroxine will eventually inhibit this if it makes too much. So whenever you have like an ending product that can turn it off, that is a great example of feedback. So if you have too much, you want to turn this off. So inhibition means turning it off. All right, and then down here, I saw this example with an AP as well. And so here's my ligand precursor. So that means it's not quite ready, but cleavage causes it to become this, and this is like active, ready, ready to work. And you know it's active, ready because it fits right in. So it fits into this receptor and it's active. But an inactive one has a different shape, which means it's not going to bind correctly. So if it isn't cut or if it isn't the same shape, then it won't work. All right. Whoops, I might have missed something at the bottom here. Nope, got it. All right, next page, cell division. All right, so we're moving on to the last part of Unit 4, which is all about mitosis. So what's the purpose of cell division? So cell division, first of all, is mitosis, and you want to do this anytime you want to replace or repair cells. Or asexual reproduction. So if you want to just make more cells, if you're a bacteria and you have one cell and you want that to divide into two cells, that's mitosis. If this cell is trying to grow, you're going to divide and make two cells. All right, draw a replicated chromosome as it appears before cell division. Label the centromere. Okay, here is my centromere. Kinetochore. Okay, kinetochore would be the little region here and here where the spindle fibers bind to it. And then sister chromatids. Here's one, here's the other. And these two are identical to each other. This was made by the process of replication. And replication happens during S of interphase. And replication we will do in our next unit, but replication is like when we have helicase and we break the hydrogen bonds and DNA polymerase says, okay, I need an A and a T and a C and a G, and it goes along and it builds so that you have these two sister chromatids which are identical to each other. The centromere holds it together, but then the spindle fibers are going to come and attach to those kinetochores to pull them apart during mitosis. All right, a chicken has 78 chromosomes. How many chromosomes did the chicken inherit from each parent? Okay, so half of 38 or 78, 39? 6, 7, yeah, 39. Whew, weird number I picked there. How many chromosomes will be in each somatic cell after mitosis? So once it is just a normal cell, it's going to have all 78. And how many are in each of the gametes? So a gamete only is going to have 39 because you put together 39 and 39 and you make 78. So gamete, like a sperm and an egg, would go together to make my 2N, my diploid cell. And how many chromosomes will be in each somatic cell of the offspring? All of the cells should have 78 in them. All right, this diagram I saw on an AP question. So amount of DNA per cell. So in G1, I have my normal. So let's do humans. So humans have 46 chromosomes. And then during S, they replicate. So that's 92 chromosomes that it would have. And then once they go through mitosis, they go back to having 46. So S, this is where DNA replication occurs. All right. Briefly describe what's happening in each phase of the cell cycle. So the cell cycle is the normal life of a cell. I always divide it up like this. G1, S, G2, and mitosis. So G1 is normal growth. If it's going to divide, it's going to increase in the amount of cyclins that it has. We'll do cyclins in a minute. S is when DNA replication occurs. G2, more growth, different cyclins, but increase in cyclins as well. M is mitosis. So this is when, whoops. Oh boy. All right, let's go back to here. Mitosis. So this is when the chromosomes are separated. They already duplicated, but now they have to separate. And cytokinesis is when the cell divides. So not the chromosome, but the actual cytoplasm and all of the organelles have to separate. All right, go through the phases of mitosis. So just the M part here. So we have prophase, metaphase, anaphase, telophase. So in prophase, my nuclear membrane is going to dissolve. So nuclear membrane dissolves. My chromosomes are going to condense, which means they thicken and I can see them. And then in metaphase, the chromatids all line up. So metaphase, you'll see it looking like this. And then anaphase, the spindle fibers pull the sister chromatids apart. So here's my sister chromatids being pulled to each side. So once they're pulled, I end up in anaphase with a cell that looks like this. You got this half, you got this half, and here's my blue spindle fibers that shortened and pulled them to the opposite sides. And then telophase, the cell separates. So we usually say that telophase and cytokinesis happen together, and it allows us to do this final step so that you have two resulting cells. All right, describe the structure and function of centrioles and MTOCs. MTOC stands for microtubule organizing center. So centrioles usually look like this. They look like literally two rigatonis next to each other, but they have a lot of microtubules in them. And so from those, spindle fibers develop. So spindle fibers are microtubules, or even microfilaments, because they're a little bit thinner and smaller, that branch out from my microtubule organizing center, which is my centriole. These also separate and go to the two poles of the cell so that they can make the spindle fibers go towards the middle. All right, how is cytokinesis accomplished in animal cells and plant cells? So in animal cells, you have what's called a cleavage furrow. So it begins to break into two by pinching in. But in a plant cell, it forms what's called a cell plate down the middle of it. And that will eventually form the wall and separate two walls, a wall for each side. All right, how is mitosis controlled? Explain the roles. Okay, so this is the really exciting AP Bio stuff. So I'm going to just draw a little picture here like I had before. So here's my G1, my S, my G2, and my mitosis. I have three different checkpoints. One is right here, one is right here, and one is here. All right, and these checkpoints, so this is my G1 checkpoint. Basically, when a cell has gone through mitosis, I am starting here with a new cell. And so my new cell, if it is a cell that wants to divide, will start increasing the amount of cyclins that it makes. And so cyclins are a protein that have a very specific shape, and they bind to cyclin-dependent kinases. So here is my cyclin-dependent kinase, and when these are bound together, they're essentially counted. And in order to pass this checkpoint right here, I need to have a high level of cyclins. And if I have a high enough level of cyclins, then I pass into S, which means I am going to start doing DNA replication. If I do not pass, I am going to go into what's called G0, which is a stage of no growth. So me at my age right now, I'm in a stage of no growth, like not getting any taller. Maybe all this staying inside is like I'm eating more, but I'm not actually growing. I'm not growing any new cells. This is something that really happens much more when you're young. My red blood cells are replacing themselves and my skin cells are replacing themselves. So they're passing on to S, but a lot of my cells are at a stage of no growth. Okay, now after a checkpoint is passed, this is a really important, I'm going to put a star. So after a checkpoint, you have to degrade your cyclins. If you do not degrade your cyclins, you're going to get back to this part again, and you're already going to have a high level. You don't want to start off with a high level. You want to start off with nothing. You want a cell to have to go through a signal transduction pathway to actually make cyclins. They have to be signaled. They have to be told, like, hey, you're a cell that should divide. I'm going to turn on your cell. You're going to activate this RNA. You're going to make the proteins called cyclins. So you want to degrade them every single time so that you don't do it on accident. All right, over here at this next checkpoint, we have a different cyclin. Its levels will also increase. It will bind to cyclin-dependent kinases, and it will allow you to pass the checkpoint. If you pass the checkpoint, you start doing mitosis. So you start doing prophase. So all of the things that make a cell in prophase, like you start breaking down the nuclear membrane, you start condensing your DNA, you start forming spindle fibers from your microtubule organizing centers, your centrioles. So if you pass the checkpoint, you go this direction. If not, you just stop, and your cell is not going to go through mitosis and not going to divide. The last checkpoint is before, so it's between metaphase and anaphase. So when the chromosomes are all lined up like this, the spindle fibers get like yanked on a little bit to make sure that they're connected to the kinetochore. And if they are, then they pass the checkpoint and keep going. So that's how cyclins work. I just want to draw a little graph to show you. If this is G1, S, G2, and mitosis, my first color will be this blue. And so this is a G1 cyclin. So I'm going to expect my G1 cyclin to go up, 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 be high enough to be counted so that I actually start dividing, and then it's going to go back down. And the reason it goes back down, so here my cyclin is going to build up, and here it's going to pass the checkpoint, and here it's going to be degraded. And then I need a different cyclin. So this is my G2 cyclin. So these levels are non-existing, and then all of a sudden during G2 they go up. So now this builds up, and then this allows me to pass my checkpoint, and then it will get degraded. And so the name cyclin actually came from the fact that it goes through a cycle. It goes up and it goes down in levels. And the control of that allows your cell to only do mitosis as desired. So increasing levels of cyclin is due to signal transduction pathways. So it will cause mitosis when desired. When desired. If you have excess cyclin, or you have no degrading of cyclin, you are going to keep doing mitosis way, way, way too much. And so they've understood that this is actually one of the things that causes cancer. When your cells are making tumors, a tumor is a mass of cells that was made too quickly, and it went through this cell cycle without hitting the checkpoints. And so there was an error in the cyclins. All right. Oh my gosh, I'm on the last page already, you guys. All right, I love this diagram because this is a great pathway that involves signal transduction, a signal transduction pathway, and mitosis control. So AP loves these diagrams. You will see this P53 molecule come up maybe even on your test this May. Who knows? So here we start off with DNA damage. I always want to find the beginning of my arrows. So here's my first arrow. If your cell has DNA damage, that is my signal. And that causes a protein kinase cascade. So that is my transduction. And my response is to activate this P53. So I know it's active because I see that phosphate group on it. I know that that is an activation signal. So my activated P53 goes and binds to DNA. So I recognize this right away as DNA by its shape. And so when something binds to it, this is my transcription factor. So an activated P53 is the transcription factor. That is going to cause transcription, which is going to make messenger RNA, which is going to go to a ribosome, and it is going to make a protein. That is called translation. There's the protein, P21. Now this protein goes, it's just in the cytoplasm. So here we are in the cytoplasm here. An active cyclin CDK complex. An active cyclin CDK complex would cause mitosis to occur. But if you bind a P21 to it, it becomes inactive. If it's inactive, you are not going to pass the checkpoint. If you don't pass the checkpoint, no mitosis. Awesome. I don't want to do mitosis if I have DNA damage. So this is such a great, great, great signal transduction pathway and an example of control because it shows how our body regulates. When there is a problem, we can shut down activities that we don't want to occur. If your DNA is damaged, let's say you were exposed to these x-rays and you had all of this DNA damage, you do not want to be going through mitosis or meiosis. Heaven forbid you're going to make gametes, and then those gametes are going to have fertilization and have a baby. So you're going to want to shut down mitosis or meiosis from occurring, and this is how it happens. This is the pathway, and you can clearly see the different steps. So I am going to give you some more practice problems on AP Classroom, and I want you to go through diagrams like this and write out like what's happening, what are the steps, what would happen if this pathway was on, what would happen if this pathway was off. All right, and then we will Zoom on Thursday, and I will go over any questions that you have. Miss you guys. Take care. Bye.